When you hear the word blueprint, you probably picture a construction site.
Right, like rolled up blue paper, white lines.
Exactly.
Yeah.
Architects pointing at steel beams. It implies organization. It implies a plan.
Yeah, totally.
But when you hear the word cancer, you picture the exact opposite. We're conditioned to picture chaos. Just this explosion of random mutated cells blindly dividing, piling up on each other, and just destroying everything in their path.
And, that chaotic, disorganized mass is historically how most of us, even within oncology, have visualized tumor growth.
Right.
The assumption has largely been that metastasis is just this anarchic, uncontrolled expansion.
But what if that assumption is entirely backward? What if the most dangerous thing a cancer cell can do isn't just dividing rapidly?
Yeah, what if it's something else entirely?
Right. What if the real reason some breast tumors stay localized and, remain totally curable while others successfully spread and conquer distant organs is because that second group of cells suddenly remembers how to read an ancient, highly organized architectural blueprint?
It's a terrifying but fascinating thought.
It really is. Welcome to the Deep Dive. Today we're exploring an absolutely fascinating 2026 paper published in the journal Cell.
Yeah, it's titled A 3D Morphogenetic Blueprint for Metastatic Outgrowth in Breast Cancer.
And the researchers here combine some wild tech, single-cell RNA sequencing, AI-supported 3D imaging, spatial transcriptomics.
Basically using all the modern tools to decode the literal 3D architectural blueprint of cancer metastasis.
It's just wild. So let's start with that genetic software, because the researchers had to figure out who the corrupted architect was.
Exactly. And the technological approach they took is what makes this so definitive. They didn't just look at the tumors.
Right.
They mapped the exact genetic software running inside the cells, visualized the physical hardware those cells built in 3 dimensions, and then identified the specific host tissue signals powering the entire construction project.
So to do that, they took fresh surgical samples, right? We're talking primary breast cancers, distant metastases that had spread to other organs, and healthy normal mammary glands, just for a baseline.
Yep. And they analyze them using single-cell RNA sequencing, or cRNA-seq.
Now, I know we throw around terms like sequencing a lot on here, but taking a tumor and trying to read its genetics is usually like throwing an entire piece of fruit into a blender.
That's a great way to put it.
You get a smoothie, and you just measure the average of what's in the glass.
Right. Bulk sequencing gives you an average. If you grind up a tumor, you're getting this messy mix of cancer cells, immune cells, fibroblasts,
endothelial cells from the blood vessel.
Exactly. You have no idea which specific cell was producing which specific RNA message. But single-cell RNA sequencing fundamentally changes the resolution.
Because they separate the fruit bowl without blending it.
Exactly. The researchers take that solid tumor tissue and use enzymes to gently dissociate it. They break it down until the cells are just floating individually in a liquid suspension.
Okay, so then what? How do you read them one by one?
So then, using microfluidics, they force these cells through microscopic tubes and trap each individual cell inside a tiny droplet of oil.
Oh, wow.
And inside that droplet is a chemical barcode. The cell is lysed-like, broken open, and its messenger RNA binds to the barcode.
And messenger RNA, that's the active instructions being read from the DNA at that exact moment, right?
You got it. So when they sequence everything later, they can trace every single active gene back to the exact individual cell it came from.
That's incredible.
And they did this for tens of thousands of cells. They were looking at the precise identity and metabolic state of every single cell in that tissue.
Okay, so they have this incredibly high-resolution map of the active text messages inside these metastatic breast cancer cells. What did the messages actually say? Because I'd assume a metastatic cell is just screaming, divide, divide, mutate.
That's what we thought. But the data showed something far more sophisticated. The metastatic cells actually retained their adult identity, specifically their transcriptome-matched adult luminal type 2, or L2 cells.
And those are the specialized epithelial cells that line the ducts in a normal, healthy breast, right?
Exactly. But overlaid on top of that adult identity, they were heavily expressing a completely different, highly coordinated set of instructions.
Wait, like a second operating system?
Basically. It was an ancient, dormant genetic program called fetal mammary morphogenesis, or FMM.
Wait, hold on. I want to make sure I'm grasping the magnitude of this.
It's big.
The cancer cell isn't just randomly mutating a new, terrifying way to spread through the body. It is literally digging into the genome's archives, pulling out an embryonic blueprint that hasn't been used since the patient was in the womb.
Yep.
and is running that software.
It is reactivating fetal development. Think about it. When an embryo is growing, it has to build complex organs from scratch. Fetal mammary morphogenesis is the specific biological program that dictates how cells grow outward, invade surrounding embryonic tissue, and form branching tubular networks.
Like roots of a tree.
Exactly. It's how our bodies build the architecture of glands, the bronchial trees of our lungs, the collecting ducts of our kidneys.
Wow.
And the researchers coined a new term for this specific hijacked program in the context of cancer. They call it Metastatic Trabecular Morphogenesis, or MTM.
Trabecular meaning a branching lattice. But, why would a cancer cell bother?
What do you mean?
Well, if its only goal is to make more copies of itself, why spend the energy to build a complex, branching fetal tube network?
It comes down to the brutal physics of survival in a foreign environment.
Okay, how's that?
If you're a single breast cancer cell that just arrived in the liver or the brain, you're in a hostile place. You have very limited access to oxygen and nutrients.
Right, you're off the grid.
Exactly. If you just divide into a solid, compact, spherical ball of cells, you create a physical barrier for yourself.
Oh, I see.
The cells on the outside of the ball might get some nutrients, but the cells trapped in the middle will quickly starve, suffocate, and die. That creates what we call a necrotic core.
So a solid ball actually limits how big the tumor can grow. It chokes itself.
It creates an immediate bottleneck. But if you activate the MTM program, you don't grow as a solid ball. You grow as a space-filling, branching network.
You weave through the host tissue.
Yes. By forming a trabecular lattice, you maximize your surface area. Almost every single cancer cell remains in direct contact with the host's blood vessels and extracellular matrix, just soaking up resources.
Okay, so if I'm tracking this correctly, and let's do a quick recap for everyone listening since we're about 5 minutes into the deep dive here.
Good idea.
Single-cell sequencing allowed the researchers to see that metastatic cells are basically chimeras. They keep their adult breast tissue disguised, but they run a fetal branching software to ensure they never choke on their own growth.
That is the core mechanism of their survival, yes.
You know, a good mnemonic for you guys to remember this is just ABC.
Right.
A for adult identity, B for branching blueprint, and C for cancer metastasis.
I like that, ADC.
And it makes you wonder, think about your own body's development. If a cancer cell can remember how it was built in the womb, what other dormant programs might it be hiding?
It's a chilling thought.
It really reframes the entire concept of a tumor. But look, reading the genetic code is one thing. If they're actually building this branching house in the lung or the brain, we need to be able to see the physical architecture.
Which is historically a huge blind spot in pathology.
Right. And the paper goes into incredible depth on how they visualize this 3D shape. Why has this been such a blind spot?
Standard clinical pathology just relies on 2D histology. A surgeon removes a piece of tissue, it gets embedded in a block of paraffin wax, and a technician uses a microtome, which is essentially a microscopic deli slicer.
A deli slicer, nice.
Right, and they cut a section that's maybe 4 or 5 microns thick. They put that single flat slice on a glass slide, stain it, and look at it under a microscope.
Which means you are only seeing a completely flat cross-section of a 3D object.
Imagine trying to understand the complex plumbing and structural engineering of an entire skyscraper by only looking at a flat floor plan of the 3rd floor.
You completely lose the z-axis.
Exactly. You miss the continuity of the structures. So to overcome this, the researchers used a deep learning AI workflow called CODA.
CODA. And they use this to reconstruct human metastases in the brain, liver, and soft tissues.
Yep.
But how does an AI turn flat glass slide back into a 3D object?
Well, they took consecutive ultra-thin slices of the tumor tissues, hundreds of them in a row. They stained them and photographed them. Okay. Then they fed all those digital images into the CODA AI. The algorithm performs complex image registration.
Meaning it lines them all up.
Exactly, but it's hard because it has to calculate affine transformations and elastic deformations. When you slice tissue that thin, it inevitably tears, stretches, or folds slightly.
Oh, sure, it's delicate.
So the AI mathematically flattens out those artifacts, aligns the cellular coordinates from slice 1 to slice 2 and slice 3, and builds a volumetric mesh. It reveals the hidden 3D lattice.
And when they finally spun that 3D model around on the screen, did it look like the fetal branching program predicted?
It matched the software perfectly, The metastases that had high expression of the MTM genetic program formed a dense trabecular lattice.
Wow.
It was a fractal, highly organized branching network of thick epithelial cords. And interestingly, unlike healthy breast tissue, which forms hollow tubes to carry milk, these cancer cords had no lumens.
So they weren't hollow.
Right. There were solid, structurally rigid branches of cells.
Okay, let me try an analogy here. If you look at a sea sponge, it's porous, it's branching, it's sprawling out in every direction to filter as much water as possible. That sounds like a trabecular tumor.
That's a great visual.
But contrast that with an expansile tumor, one that just grows as the solid mass. An expansile tumor would be like a potato, a compact, smooth ball that just gets bigger by physically pushing the surrounding dirt or tissue out of the way.
The sponge versus the potato is a perfect starting point, but we have to refine it slightly. A sea sponge is a passive filter. These trabecular networks are active and aggressive.
Okay, how so?
They aren't just growing into empty space, they're actively dissolving and remodeling the host organ to make room for those solid branches. But your morphological contrast is totally correct.
And did they prove this was specific to breast cancer cells?
They did, through rigorous control experiments in mice. They injected different types of breast cancers, but also fibrosarcomas and osteosarcomas.
And those sarcomas derive from connective tissue and bone, right? Not epithelial duct.
Exactly. And the sarcomas grew in the lungs and brains of the mice as those smooth, compact potatoes. They were purely expansile. Only the breast cancer cells formed the branching sponge, proving this trabecular blueprint is a specific morphogenetic trick tied to their epithelial origin.
Yeah, here is the clinical dilemma that jumps out to me. If you're an oncologist or a pathologist in a community hospital, you don't have an AI supercomputer to run code EI on every patient's tumor.
No, definitely not.
You have a microscope and a glass slide. So how on earth do you spot a 3D fractal sponge when you're stuck looking at a 2D slice?
The researchers solved this brilliantly. They applied a mathematical concept to the pathology slides called topomorphometric parameters.
Okay, that's a mouthful.
It relies on the geometry of fractal dimensions. Think about slicing straight through that potato you mentioned. Your cross-section is going to be one giant solid circle of tissue.
Right.
Mathematically, that shape has a very low perimeter to area ratio. It's mostly inside, with very little edge.
Okay, that makes sense. A solid mass gives you a solid circle, low perimeter, high area.
But if you take a knife and slice straight through a complex branching sea sponge, you don't get one big circle. Your 2D slice will look like dozens of tiny scattered islands of tissue separated by empty space where the branches weren't touching.
Oh, I see.
And all of those tiny individual islands have a massive amount of edge. They have a staggeringly high perimeter to area ratio.
So a pathologist doesn't need to see the 3D structure. They just need to look at the 2D slide and ask, am I looking at one giant continent of cells or an archipelago of tiny islands?
Exactly. That geometric signature is the proxy for the 3D architecture. And to prove its clinical relevance, they analyzed a real-world cohort of 132 patients with triple-negative breast cancer, or TNBC.
And triple-negative is notoriously aggressive.
Very aggressive. They went back and looked at the original biopsy slides of the primary tumors in the breast before the cancer had ever spread.
Okay, and what did they find?
They calculated the perimeter to area ratio of the tumor islands. The prognostic power was staggering. The primary tumors that had an expansile architecture, the solid, potato-like continents, tended to stay localized. Those patients had much higher rates of being cured after surgery.
But the islands?
The primary tumors that already exhibited a trabecular architecture, the scattered islands with the high MTM signature, those were the ones that relentlessly metastasized to the brain, lungs, and liver.
Wow. So to do a quick 10-minute recap for you guys listening. CODA AI reveals metastases grow as branching trabecular lattices, not solid spheres.
Right.
And 2D slice patterns, like looking for many islands and a high perimeter, can predict that 3D structure, meaning trabecular primary tumors spread and expansile ones don't.
Exactly.
So think about it. If you were a pathologist looking at a slide right now, could you spot the difference between the sponge islands and the potato mass? Because the cancer is essentially practicing its architecture in the breast.
Yes. It's building the fractal branching network locally, and if it figures out how to do that, it's primed to do it anywhere in the body.
If I'm an oncologist, knowing whether I'm looking at an expansile mass or a trabecular lattice completely changes my risk assessment for that patient.
It changes the entire paradigm of how aggressively we might need to treat them post-surgery.
Okay, so we've established the blueprint, which is the MTM program. We can visualize the physical house, the trabecular sponge.
Yep.
The obvious next question is, who is running the construction site? I mean, the DNA might contain the blueprint, but DNA doesn't do anything by itself. It needs something to read the instructions and force the cell to build.
You're asking about the regulatory mechanism, and to find the master switch, the researchers had to map the epigenome.
Now, for those of us who need a quick refresher, epigenetics goes beyond just the sequence of the DNA letters, right?
Yes. Every cell in your body has the same DNA, the same hardware. But a liver cell looks different than a brain cell because of the epigenome, the chemical modifications that sit on top of the DNA and dictate which genes are accessible and which are locked away.
Like little tags.
Exactly. DNA is wrapped around proteins called histones. If the histones are packed tightly, the genes are hidden. If they're loose, the genes can be read.
So how do you figure out which genes are loose and active in these specific metastatic cells?
They grew metastatic patient-derived organoids, which are basically three-dimensional mini-tumors grown in a dish from real patient samples.
Okay.
Then they used a technique called CHIP-T-Sec. This allows researchers to use antibodies to grab onto the specific histone proteins that are marked with active chemical tags and pull down the DNA attached to them.
And then they fed this data into a machine learning algorithm, right? From HMM.
Yes, to map out the entire regulatory landscape.
They're essentially dusting the genome for fingerprints to see which switches have been flipped.
Exactly. They were specifically hunting for active enhancers and promoters.
What's the difference there?
Promoters sit right next to a gene and act as the direct ignition. Enhancers can be located thousands of base pairs away on the DNA strand, but the chromatin literally loops back on itself in 3D space, so the enhancer can touch the promoter and supercharge the gene's expression.
Oh, that's wild. The DNA literally bends to touch itself.
Yep. And when they mapped the chromatin looping for the MTM branching genes, they wanted to see what was sitting at the controls.
Yeah, what was it?
They found that the regulatory regions driving this branching morphogenesis were overwhelmingly bound by a specific family of transcription factors, ETV1, ETV4, and ETV5.
ETV. So these proteins are the foremen of the construction site. They're the ones reading the MTM blueprint and telling the cell to start branching.
They are the master regulators, and the researchers proved this with a brilliant CRISPR experiment in mice.
Let's hear it.
They took metastatic breast cancer cells and used CRISPR gene editing to surgically delete the ETV genes. They took normal, wild-type cancer cells and labeled them with a green fluorescent protein.
Okay, green is normal.
Right. And they took the ETV knockout cells and labeled them with a red fluorescent protein. They mixed them together and introduced them into the mice to observe the physical differences in how they grew.
Okay, I read this part of the source material, and I have to push back on something because it seems completely counterintuitive.
Oh.
You knock out the master regulatory switch for metastasis. You'd assume the cancer is completely crippled. But the paper explicitly notes that the primary tumor in the breast still grew perfectly fine without ETV.
That's right.
The red cells and green cells divided equally. Even more confusing, single cells from both the red and green groups successfully escaped into the bloodstream and traveled to the lungs.
Yep.
So if ETV is the master switch for metastasis, why didn't deleting it stop the cells from spreading?
That is the most elegant mechanical insight in the entire study. It comes down to space constraints and tissue resistance. Think about where the primary tumor is growing.
In the breast.
Right. The mammary fat pad is a relatively permissive, flexible environment. A cancer cell does not need a sophisticated architectural strategy just to divide and create a local mass there. And it certainly doesn't need ETV to simply float passively through the bloodstream.
Well, yeah, floating doesn't require construction.
Exactly. But arriving in a distant organ is a totally different mechanical challenge. The lung or the brain is a dense, highly structured foreign environment. You cannot just passively divide.
You just get squished.
The physical resistance of the host tissue will crush you, or you will starve. To survive, you have to actively remodel the extracellular matrix. You have to secrete proteases to dissolve the host tissue and physically push outward in that branching, space-filling pattern to reach the blood supply.
And that physical remodeling requires the ETV foremen.
Yes. Without ETV, the red knockout cells reached the lung and failed to build. They just sat there in the tissue as dormant microscopic dots. They couldn't overcome the physical constraints of the lung.
But the green ones.
Meanwhile, the green wild-type cells with the ETV foremen successfully executed the branching morphogenesis, remodeled the lung tissue, and grew into massive, lethal metastases.
That is profound. The cancer cell can successfully complete the entire journey. It can mutate escape the breast, survive the bloodstream, and land in the lung. But if it cannot physically build its 3D infrastructure once it arrives, it's completely neutralized as a threat.
It's like an invading army arriving on a hostile beach with no engineers and no supplies. They just sit there.
Okay, time for our 15-minute recap. So, epigenetic mapping found ETV1, 4, and 5 act as master switches for the MTM program. Knocking out ETV stops large metastases from forming, but doesn't stop the primary tumor from growing or traveling.
Exactly.
A quick mnemonic for this is TEC. PEC. Trabecular architecture requires ETV to build cords.
TEC, I love that.
And so to everyone listening, consider the vulnerability here. If a cancer cell travels to your lung but can't build its house because it lacks the ETV foramen, is it truly a threat?
It changes how we define the threat entirely.
Which brings us to the ultimate clinical goal. If the structural integrity of the tumor is its weakness, how do we target it? Because we can't easily use CRISPR to edit the genes inside a living human patient's entire body.
No, we definitely can't.
And if ETV is inside the cell's nucleus, it's notoriously difficult to target with a drug. So how do we stop the construction project?
The researchers had to look outside the cancer cell. They had to look at the neighborhood. To do this, they utilized a cutting-edge technique called spatial transcriptomics.
Now wait, how does that differ from the single-cell RNA sequencing we discussed earlier?
Yeah.
I thought scaRNA-seq already gave us the ultimate resolution.
Single-cell sequencing gives us incredible detail about what is happening inside the cells, but because we had to dissolve the tissue into a liquid to run the machine, we lose all the spatial coordinates.
Oh, because it's a smoothie again.
Right. We don't know where the cells were sitting relative to each other. Spatial transcriptomics solves this. Researchers place a thin slice of the tumor onto a specialized glass slide that's printed with millions of microscopic spots. Each spot contains unique DNA barcodes that act like spatial zip codes.
So it's basically a microscopic grid map.
Yes. When the tissue is placed on the slide and permeabilized, the RNA from the cells drops straight down and binds to the zip codes directly beneath them.
Oh, yeah.
So when you sequence it, you can overlay the gene expression data directly onto the high-resolution microscope image. It's Google Maps for gene expression. It tells us exactly what instructions are active and exactly where they are happening physically in the tissue.
And what did this Google map reveal about the neighborhood around these metastatic tumors?
It revealed that the cancer cells weren't building this lattice in isolation. The MTM-high cancer cells at the very edges of the branching network were receiving a massive influx of chemical signals from the surrounding host tissue, the stroma.
Wait, the healthy host organ is helping the cancer build its house.
Inadvertently, yes. The host tissue, particularly the fibroblasts, was secreting a protein called fibroblast growth factor, or FGF.
Okay.
And the cancer cells at the invasive front were densely studded with receptors called FGFR fibroblast growth factor receptors. The FGF from the host plugs into the FGFR on the cancer cell.
And let me guess, that signal travels down to the nucleus and activates the ETV foramen.
You've got it. The host provides the raw signal, the receptor catches it, the ETV foramen are activated, and the MTM branching blueprint is executed.
That's insidious.
What's darkly brilliant about this is that FGF signaling is the exact same pathway the developing embryo uses to stimulate normal lung and gland branching. The cancer has quite literally hacked the host organ's normal communication infrastructure.
But knowing that it relies on a signal from the outside is a massive vulnerability. We might not be able to target the ETV foramen inside the nucleus, but we can absolutely block the FGFR antennas on the surface of the cell.
We can, and we already have the tools to do it. The researchers tested this by using an FDA-approved drug called erdafitinib.
Erdafitinib?
Yes, which is an FGFR inhibitor already used in certain types of bladder cancer. When they treated the mice with erdafitinib, the cancer cell's antennas were blocked. They couldn't hear the FGF signal from the host organ.
And without this signal,
the metastatic burden plummeted. The cells arrived in the lung, but they couldn't trigger the ETV foramen. They lost the ability to build the trabecular lattice, and they remained trapped as microscopic, harmless clusters.
That is a staggering finding. So, to recap this final piece, spatial transcriptomics shows host tissues feed FGF signals to tumor FGFRs. And blocking FGFRs stars the tumor of the signal needed to branch, halting macroscopic metastasis.
It stops it in its tracks.
So here's a question for you listening. We are essentially turning the host organ's own communication lines against the cancer. How might this change the way we treat breast cancer patients before metastasis even appears? I mean, if FGF signaling is a fundamental pathway that normal cells use to communicate and maintain tissue health, doesn't turning it off with a drug like erdafitinib cause significant side effects for the patient.
That's the challenge. It's a systemic therapy, and yes, FGFR inhibitors do have side effects, particularly regarding phosphate levels in the blood, skin issues, and eye toxicity.
Right, because you're blocking normal cell chat, too.
Exactly. But the implications for how we sequence treatments are massive. If we can identify a primary breast tumor that has that high-risk trabecular fractal architecture on a 2D slide, we know those cells are primed to metastasize.
We can be proactive.
Right. Could we utilize an FGFR inhibitor in the adjuvant setting, meaning right after surgery, to ensure that any microscopic cells that escaped into the blood are permanently starved of the signals they need to colonize a new organ?
We're essentially turning the host organ's communication lines off, rendering the cancer deaf and structurally impotent.
Exactly.
So if we synthesize everything we've unpacked today from this cell paper, the central dogma of metastasis has to change. It is not a random, chaotic explosion of mutated cells.
Oh, no, not at all.
That old textbook image is fundamentally incomplete. Metastasis is an incredibly organized, highly structured 3D morphogenetic process. These cells survive by hijacking a dormant fetal blueprint.
They deploy epigenetic master switches.
And they steal signals from our own healthy tissues to build a highly efficient, space-filling fractal lattice.
And if we step back and look at the broader biological implications of this, it leaves us with a truly provocative thought.
Which is?
If a metastasis behaves by building highly organized fractal structures that actively remodel their environment, communicate with surrounding cells, and maximize resource extraction, perhaps we need to stop thinking of cancer simply as a tumor or a dumb mass of rogue cells.
Oh, wow. I see where you're going. If it is a blueprint, an infrastructure, and a communication network, we should start treating cancer as a rogue, rapidly developing organ in its own right.
Precisely. It is a rogue organ, and if it's an organ, its reliance on infrastructure is its ultimate Achilles heel.
That makes total sense.
Because a chaotic explosion is very hard to stop, but a highly orchestrated construction project. Once you understand the blueprints, and you know who the foremen are, you know exactly where to hit it to bring the whole building down.
A rogue organ. That is a paradigm-shifting way to look at this disease. And it's going to completely change how I read the next headline about oncology.
Take two.
Think about that the next time you hear about cancer research.
Yeah.
It's not just chaos, it's corrupted architecture. And we finally have the tools to read the blueprints. Thanks for taking the deep dive with us.